In a quiet corner of West Virginia lies Herwood, a historic cemetery tied to the Washington family. For decades, historians and descendants wondered about the exact resting places of George Washington's relatives, but the records were incomplete. In 1999, a team of archaeologists began a careful investigation at the site, hoping to confirm a location of Samuel Washington's grave. Using ground-penetrating radar, they detected rectangular anomalies beneath the surface that suggested burial shafts. The team knew that some remains had been moved in the late 19th century during an organized transfer of graves, but it was unclear how complete the previous removal had been. As the soil was carefully scraped and excavated, small fragments of bone began appearing in clusters. Some were only an inch long, others larger but worn and fragile. Each fragment was treated with painstaking care. They were mapped, photographed, labeled, and stored, providing a clear record for future study. Even though these bones were small remnants, they represented lives that had once been fully interred at the site. Historians knew that this first step, simple as it seemed, might hold the key to uncovering long-forgotten secrets about one of America's most famous families. These fragments were tiny pieces of a puzzle that modern science could one day solve. While the excavation concluded without immediate answers, the bones were preserved meticulously, stored under controlled conditions, and recorded with every relevant detail. Little did anyone know that decades later, advancements in DNA technology would transform these small fragments into a groundbreaking revelation about the Washington family's heritage. This moment highlights how careful fieldwork, combined with future scientific innovation, can uncover historical truths that documents alone cannot reveal. Decades after the excavation, the preserved fragments were sent to a laboratory specializing in ancient DNA analysis. The team faced a major challenge. The bones had been underground for over a century. Time, moisture, and bacteria had all contributed to the degradation of genetic material. Most standard DNA tests were ineffective on remains this old. Human DNA was present, but in extremely small amounts, often overwhelmed by bacterial contamination. To overcome this, researchers used advanced methods designed for degraded samples. They focused on 3 types of genetic markers: Y chromosome DNA, which traces a paternal line; mitochondrial DNA, inherited through the maternal line; and autosomal SNPs, which scatter across all chromosomes and help determine overall family relationships. These tiny fragments of DNA were like pieces of a shattered puzzle. The laboratory team worked slowly and carefully, using techniques to separate human DNA from bacterial material and capture sequences specific to the Washington family. Each step required precision, as rushing could destroy the limited genetic material. Modern sequencing machines, capable of reading even tiny fragments only a few dozen base pairs long, allowed the team to recover thousands of tiny reads. These reads were then aligned and filtered to remove errors and contamination. Analysts had to spend hours reviewing each sequence, confirming whether it belonged to a human or a foreign organism. Collaboration with genealogical experts added further insight, helping interpret the genetic patterns and match them with historical records. The work required patience, skill, and meticulous attention to detail. This process demonstrates the complexity of working with centuries-old DNA and shows how modern technology can unlock secrets hidden in the soil for generations. Once the genetic data were carefully processed, researchers began comparing it to what was already known from historical documents. Cemetery records from the 19th century noted partial disinterments, moved headstones, and unmarked graves. Many graves have been disturbed during the 1882 reorganization of the Washington family plot, leaving only fragments behind. By comparing DNA profiles with these records, scientists could start connecting the fragments to specific family members. Y chromosome markers confirm paternal lines, mitochondrial DNA trace maternal ancestry, and autosomal SNPs establish broader kinship relationships. Every type of data worked together to create a reliable picture of the family structure. This combination of genetic analysis and historical documentation allowed researchers to identify the remains of 3 key individuals: Samuel Washington, George Steptoe Washington Jr., and Lucy Payne Washington. These identifications were not just scientific accomplishments. They revealed a hidden chapter of the Washington family story. For centuries, Samuel Washington's resting place had been unknown, obscured by time and incomplete records. Modern DNA methods, combined with meticulous excavation and archival research, finally provided clear answers. The study showed how fragmentary remains, when carefully analyzed, can reveal detailed family connections, giving both historians and descendants new understanding of the past. This integration of science and history demonstrates the power of DNA analysis to complement traditional archival research, filling in gaps left by centuries-old records. After the fragment arrived at the specialized DNA laboratory, the focus shifted from archaeology to forensic science. The preserved bones, carefully labeled and boxed, were now handled under strict laboratory protocols designed to prevent contamination. The lab team worked in controlled environments with filtered air and sterile equipment to protect the delicate samples. This was essential because even tiny modern DNA traces could overwhelm the century-old genetic material. Each fragment was examined to assess its condition, noting breaks, soil compression, and signs of previous handling. The technicians knew that conventional DNA profiling methods would not work for remains this degraded. Instead, they relied on targeted capture techniques designed to extract the tiniest fragments of human DNA from the surrounding material. By isolating Y chromosome sequences, mitochondrial DNA, and autosomal SNPs, they could build a comprehensive genetic profile, despite the challenges. Each extraction cycle was repeated multiple times to maximize recovery, and sequencing machines capable of reading only a few dozen base pairs were employed to ensure accuracy. Every step was slow, meticulous, and carefully documented. Analysts spent hours aligning the captured DNA fragments, filtering out environmental contamination, and separating human sequences from bacterial or fungal DNA. Collaboration with genealogical researchers allowed the team to cross-reference findings with known Washington family lineages. The lab work required patience, technical skill, and rigorous attention to detail, but provided a strong foundation for the next stage, connecting genetic data to historical evidence. This stage demonstrated how modern forensic methods can solve mysteries that archival research alone could never resolve, and it highlighted the extraordinary care needed to handle fragile, centuries-old remains. It was at this stage that the hidden Washington family secrets began to take shape, revealing connections that had been lost to time. The scientists relied on 3 types of DNA markers to establish family relationships. First, the Y chromosome markers trace a paternal line from father to son, helping identify male members of the Washington family. These sequences are passed nearly unchanged from generation to generation, making them powerful tools for confirming ancestry. Next, mitochondrial DNA trace a maternal line, which passes from mother to children. This allowed the researchers to verify connections on the maternal side, particularly in cases where female family members had been previously unrecorded or misidentified. Finally, autosomal SNPs provided a broader perspective. These genetic markers are spread across all chromosomes, reflecting contributions from both parents and extended family lines. By analyzing tens of thousands of these markers, researchers could estimate kinship with great precision, determining how closely individuals were related across multiple generations. Each type of DNA data played a specific role, but together they created a comprehensive picture of the family structure. The results confirmed both maternal and paternal relationships, revealing a consistent pattern that matched historical records from Herwood Cemetery. This dual approach, combining Y chromosome, mitochondrial DNA, and autosomal SNPs, was essential because the fragments were incomplete and heavily degraded. By layering multiple lines of evidence, the scientists reduced the risk of errors and ensured that the identifications were reliable. It also meant that the discoveries were backed not just by probabilities, but by multiple independent measures, providing confidence in the conclusions. This careful methodology turned tiny, fragmented bones into powerful sources of historical insight, demonstrating how modern genetics can illuminate the past with unprecedented clarity. Once the lab established reliable DNA profiles, researchers began the crucial step of connecting the fragments to specific Washington family members. Historical records provided context, noting that the cemetery had been disturbed in 1882 and that only partial remains remained in certain locations. The Y chromosome markers confirmed the paternal line of male family members, while mitochondrial DNA highlighted maternal connections, particularly to Lucy Payne Washington, who acted as a matriarch in the family branch. Autosomal SNPs filled in gaps, confirming generational relationships and broader kinship patterns. By carefully comparing DNA data with archival records, researchers successfully identified 3 individuals: Samuel Washington, George Steptoe Washington Jr., and Lucy Payne Washington. Samuel Washington, George Washington's younger brother, had rested in an unmarked grave for over a century, his location obscured by both incomplete historical records and fragmentary remains. George Steptoe Washington Jr. and Lucy Payne Washington were also identified, completing a small but critical family group. These findings solved long-standing mysteries, confirming the final resting places of prominent family members and providing living descendants with concrete evidence of their lineage. The combination of DNA and historical documents created a 2-track validation system, ensuring that the identifications were accurate and supported by multiple lines of evidence. The study demonstrated how even fragmentary remains, previously thought too degraded to analyze, could reveal precise family relationships with modern methods. This achievement highlighted the power of integrating science with history and genealogy, showing how centuries-old secrets can finally be brought into the light. The DNA study had profound implications for the descendants of the Washington family. For generations, many had known that some relatives were buried in Herwood Cemetery, but exact locations remained a mystery. Family lore, wills, and estate records often contradicted one another, leaving gaps in the lineage that could only be speculated about. With the results of the DNA testing, descendants now had definitive evidence of their connections to historical figures. Samuel Washington's resting place was confirmed, and living descendants could finally reconcile the family history that had long been obscured. George Steptoe Washington Jr. and Lucy Payne Washington were also accurately placed within the family tree, providing further clarity. For some descendants, this knowledge had emotional significance. Being able to connect the tiny fragments of bone to ancestors brought a tangible sense of heritage and belonging. Private family records, some maintained for decades, were updated to reflect these verified relationships. Descendants could also trace paternal and maternal lines with precision, thanks to the Y chromosome and mitochondrial DNA analysis. The study provided not just historical confirmation, but also a personal connection to a past that had been physically and metaphorically buried for over a century. This demonstrates how modern science can directly impact living families, allowing them to uncover the personal stories behind historical figures. Beyond just names and dates, the DNA study allowed the family to see the relationships, generational spacing, and maternal-paternal connections in a way that no written record could fully convey. It is rare for science to resolve such long-standing genealogical mysteries, but the Herwood case shows the potential of integrating DNA analysis with historical research to uncover family truths that have been hidden for generations. While the findings were personally significant for descendants, they also offered new insights for historians. The Herwood Cemetery had long been known to contain Washington family members, but records of disinterments, partial relocations, and unmarked graves had made it difficult to understand who was buried where. Historical documents from 1882 describe headstones being moved, graves partially disinterred, and remains being relocated to new plots. Some fragments were overlooked during this process, left behind in soil pockets or mixed with debris. This created a patchwork of physical evidence that did not always match the written records. The DNA study provided a bridge between what had been recorded and what physically remained. By mapping the recovered bone fragments and comparing them to historical documents, researchers could confirm the location of previously unmarked graves. This alignment of genetic data and archival research offer a level of historical clarity rarely possible in family cemeteries disturbed over a century ago. Historians could now anchor decades of speculation in hard evidence, understanding exactly which family members rested in specific locations. The study also highlighted how cemetery practices, inheritance disputes, and family decisions influence burial arrangements. While Samuel Washington was historically significant as George Washington's younger brother, the reason for his unmarked grave could be traced to both practical and personal factors. The combination of archival records and DNA evidence revealed that these decisions had long-lasting effects on the preservation of family history. The findings underscore the importance of interdisciplinary research, where history, archaeology, and genetics work together to reveal stories that would otherwise remain buried in silence. The Herwood Cemetery Project also has broader implications for genealogy and historical research. Traditionally, genealogists relied on wills, land records, and estate maps to track family connections, but these sources are often incomplete or inaccurate. Unmarked graves, like those of Samuel Washington and his relatives, frequently left gaps in the historical record. DNA analysis provides a reliable method to confirm or challenge assumptions about family relationships, especially in cases where historical documentation is fragmented. By combining Y chromosome data, mitochondrial DNA, and autosomal SNPs, researchers can establish direct paternal, maternal, and overall familial connections with high confidence. This approach is not limited to the Washington family. It can be applied to other historical families whose records are incomplete, or to communities affected by cemetery disturbances, migrations, or historical events. The Herwood study demonstrates that even heavily degraded or fragmentary remains can yield conclusive results when modern DNA techniques are applied carefully. For genealogists, this means that previously unsolvable puzzles can now be approached scientifically. For historians, it shows how DNA analysis can complement archival research, providing physical evidence to confirm the written record. This combination strengthens the accuracy of historical narratives and allows for a more complete understanding of family histories, inheritance patterns, and cultural practices across generations. The identification of Samuel Washington and other relatives also raises broader questions about hidden graves and family secrets on historic estates. How many unmarked graves remain undiscovered, waiting for modern science to uncover them? In many old family cemeteries, partial disinterments, inheritance disputes, and shifting property boundaries left fragments behind, creating mysteries that endure for generations. The Herwood case shows that these gaps in the historical record are not just academic, they have personal and cultural significance. Unmarked graves may conceal stories of prominent families, forgotten members, or overlooked children, each with a role in the broader history of a lineage. The discovery at Herwood also emphasizes the ethical responsibility of archaeologists and geneticists to preserve and analyze remains carefully. Without meticulous excavation, recording, and storage, the information needed for later DNA analysis could have been lost forever. Modern genetics provides the tools to bring these hidden stories to light, but only if past and present work is coordinated. In this sense, the Herwood study is not just about the Washington family. It is a blueprint for how researchers can approach other historic cemeteries and burial sites, uncovering human stories that have remained silent for centuries. The Herwood DNA study demonstrates how modern science can illuminate the human side of history. By recovering and analyzing fragile, century-old bone fragments, researchers not only solved genealogical mysteries, but also revealed the private lives and burial practices of a prominent family. The combination of Y chromosome, mitochondrial DNA, and autosomal SNP analysis provided an unprecedented level of certainty, confirming paternal, maternal, and overall family connections. For scientists, the study is a remarkable example of interdisciplinary collaboration, showing how archaeology, genetics, and archival research can work together to reconstruct a family tree that was once fragmented and incomplete. Beyond the technical achievements, the study also offers cultural insights. It reminds us that history is not just a story of public figures, dates, and events. It is also the story of families, personal decisions, and the lives that continue quietly beneath the soil. Samuel Washington's unmarked grave, for instance, illustrates how even historically important individuals can be forgotten by the public record, leaving traces only in fragmentary remains and oral histories. The study encourages reflection on how families and societies preserve memory, honor ancestors, and manage lineage across generations. It also shows the value of scientific rigor and careful documentation, emphasizing that discoveries of this kind are only possible when researchers treat both the material and historical evidence with respect and precision. The success of the Herwood Project has far-reaching implications for future historical and genealogical research. Techniques used in this study can be applied to other historic families, cemeteries, or archaeological sites where records are incomplete, graves are unmarked, or remains are degraded. Y chromosome analysis allows researchers to trace paternal lines. Mitochondrial DNA reveals maternal connections, and autosomal SNPs provide a detailed map of overall familial relationships. These methods give both historians and genealogists tools to confirm or correct historical narratives, providing tangible evidence where speculation was once the only option. The study also sets a standard for preservation, excavation, and laboratory handling of delicate remains, ensuring that critical information is not lost over time. Importantly, the Herwood case illustrates the value of combining multiple sources of evidence, archival documents, physical remains, and genetic data, to produce a reliable, verifiable understanding of family histories. This approach can be replicated globally, offering potential to uncover lost histories in communities affected by migration, war, or natural decay. Researchers now have a roadmap for integrating science and history, showing that even incomplete or fragmentary evidence can yield meaningful insights when examined carefully. Ultimately, the DNA analysis of the Herwood Cemetery fragments transformed what was once a historical mystery into a documented family story. The study confirmed the identities of Samuel Washington, George Steptoe Washington Jr., and Lucy Payne Washington, connecting living descendants to their ancestors and providing clarity for historians. Beyond individual identifications, the project demonstrates the broader potential of modern science to uncover forgotten stories buried in soil and time. It highlights the importance of careful excavation, meticulous documentation, and the application of advanced genetic techniques to historical research. More broadly, the study reminds us that history is not only recorded in books, but is also embedded in the physical traces left by past generations. Each grave, fragment, and genetic marker carries a story waiting to be told. For the Washington family, descendants, and the public, the Herwood discovery enriches understanding of both the private and public lives of one of America's most iconic families. It also serves as a model for future investigations, inspiring researchers to explore hidden histories, fill gaps in records, and bridge the centuries between the past and present. In the end, science did more than solve a mystery. It revived a part of history that had been silent for over 100 years, allowing voices from the past to be heard once again. In a quiet cemetery in West Virginia, scientists recently uncovered long-forgotten secrets of George Washington's family. Small bone fragments, carefully preserved for decades, reveal connections that have been lost to history. Modern DNA analysis, combined with archival research and meticulous excavation, brought clarity to unmarked graves, identifying family members whose final resting places had remained a mystery for over a century. This discovery highlights how science can illuminate both the private and public sides of history. The story began at Herwood Cemetery in Charlestown, West Virginia, during an excavation in 1999. The team used ground-penetrating radar to locate graves tied to Washington family members. Early scans revealed rectangular shapes in the soil, indicating burial shafts, but no one could know exactly how many individuals remained or how complete their remains would be. As digging began, small clusters of bones appeared, some only an inch long, others slightly larger, but all were worn and fragmented. Historical records indicated that in 1882, marked graves had been relocated, but not everything was moved. Some graves had collapsed, coffins had deteriorated, and fragments were left behind. Every bone recovered in 1999 was carefully photographed, mapped, and cataloged. Though incomplete, these remains became a treasure trove for future research. The excavation illustrated the challenges of reconciling historical records with physical evidence and set the stage for the application of modern DNA techniques decades later. Once recovered, the bone fragments were stored under controlled conditions to prevent further degradation. In the late 1990s, DNA technology was not advanced enough to extract meaningful results from such tiny and heavily degraded samples. Decades later, modern sequencing methods finally allowed scientists to revisit the fragments. When the remains arrived at a specialized laboratory, the focus shifted from archaeology to forensic precision. Technicians worked in sterile, controlled environments to prevent contamination, isolating DNA fragments from bacterial or fungal material that had colonized the bones over time. Every step, from extraction to sequencing, was conducted slowly and methodically. The team used advanced techniques to capture Y chromosome sequences, mitochondrial DNA, and autosomal SNPs, building comprehensive genetic profiles despite the age and fragility of the samples. Collaboration with genealogists and historical experts ensured that every result was cross-referenced with known Washington family lineages. The discovery had profound implications for living descendants. For decades, many had speculated about missing relatives, but now they had definitive proof. Samuel Washington's unmarked grave, along with the other identifications, allowed descendants to reconcile their family stories. They could trace paternal and maternal lines with accuracy, thanks to Y chromosome and mitochondrial DNA. This connection was more than historical. It was deeply personal, giving families a sense of closure and belonging that only modern science could provide. The study also provided valuable insights for historians. The Herwood Cemetery had been partially disturbed in 1882, leaving fragmented remains. By aligning DNA results with archival records, researchers could confirm which family members were buried where. The findings clarified burial practices, inheritance decisions, and family dynamics that had long been obscured. Samuel Washington's unmarked grave illustrated how even prominent historical figures could be lost to incomplete records and disturbed cemeteries. The study demonstrated the power of combining archaeology, history, and genetics to reconstruct events that had remained hidden for over a century. The Herwood study has far-reaching implications for genealogy and historical research. Unmarked or fragmented graves are common, and historical records often leave gaps in family histories. By applying modern DNA techniques, researchers can confirm or challenge assumptions, even with degraded remains. The study provides a model for future investigations into other historic families or communities affected by migrations, cemetery disturbances, or natural decay. It demonstrates that even incomplete evidence can reveal clear and meaningful family connections when approached carefully and scientifically. The discovery of Samuel Washington and others also raises questions about hidden graves elsewhere. How many other unmarked or forgotten burials exist, waiting for modern science to reveal them? The Herwood Project highlights the ethical responsibility of researchers to preserve and study remains carefully. It also underscores the cultural significance of burial practices and the stories they contain. Beyond genealogy, the study encourages reflection on how societies preserve memory and honor ancestors, and how science can uncover stories that have been silent for centuries. Ultimately, the Herwood DNA study bridged past and present, revealing family connections that have been lost for more than a century. Samuel Washington, George Steptoe Washington Jr., and Lucy Payne Washington were finally identified, connecting living descendants to their heritage. The study highlights the power of interdisciplinary research, combining archaeology, genetics, and archival work to solve historical mysteries. It also demonstrates that history is not only written in documents, but embedded in the traces left by past generations. For the Washington family, descendants, historians, and the public, the findings enrich our understanding of both private and public histories. Science, meticulous research, and modern technology have revived stories that were once silent, showing that even the smallest fragments can carry the weight of history. Hidden in a quiet West Virginia cemetery, forgotten fragments of George Washington's family waited for over a century, until modern DNA analysis revealed secrets that had been buried, literally, under the soil. What we uncovered connects living descendants to their ancestors and reshapes history itself. The story begins in 1999 at Herwood Cemetery in Charlestown. Scientists used ground-penetrating radar to locate graves tied to Washington family members. Early scans showed rectangular shapes in the soil, likely burial shafts. But how many individuals remained? And what condition were the remains in? As the soil gave way, tiny bone fragments appeared, some only an inch long, others slightly larger, but all worn and fragile. Historical records suggested that in 1882, some graves were relocated. Yet, fragments were left behind. Each piece was meticulously mapped, photographed, and stored, preserving them for future discovery. Decades later, technology caught up. Fragile fragments arrived at a specialized lab, where the work shifted from archaeology to forensic precision. Every step, from DNA extraction to sequencing, was carefully controlled to prevent contamination. The team targeted 3 types of DNA markers: Y chromosome for paternal lines, mitochondrial DNA for maternal links, and autosomal SNPs for broader family connections. These markers allowed researchers to identify three key family members: Samuel Washington, George Steptoe Washington Jr., and Lucy Payne Washington. Samuel, George Washington's younger brother, had rested for over a century in an unmarked grave,